Thank you. And next up is Kenny Baillie. And we're just going to set up here, so it may take a minute or two.
So if that's broadcasting here. Is it broadcasting on Zoom? I'll just go to full screen.
Yeah. So people online, can you see Kenny Baillie's?
This sofa.
So
Oh, so we need to switch.
So which one am I?
Oh, this is
that one.
that's the
Okay.
wonderful. Thanks.
Do you want to introduce yourself?
Yeah, I've wasted enough time. So thanks very much indeed for the invitation to talk. It seems a bit disrespectful to load my slides right before speaking. I was banned from entering the building, even though I technically work here, due to a mild misunderstanding. And I do consider it a privilege to address this audience, and I'll tell you why. I've been peripherally involved in Chrissy's efforts to study long COVID using genetics for a while, and it has really been an education and inspiration for me because of the exemplary public engagement that Chris and the whole team achieved. I thought we were doing that well in the study that I'm going to tell you about, but I was wrong. The direct involvement of patients and the public in every step and every decision is really fantastic, and of course leads to better science. That's obviously what we all want. So I'm delighted to tell you about our work on acute COVID-19 here in Edinburgh and across a large part of the world now. I'll explain the findings of the study, and I know there are obviously lay members in the audience, but I think that in particular I now know from interacting with so many of you that this particular group of lay people are very well informed in scientific and biological topics. So please let me know if I skip over anything that you would want to have explained. But I'll pitch my talk at the level that I usually do when I speak to clinicians who don't do this kind of science, but are obviously trained in biology. So our public engagement was really through an interaction with this charity. The tiny logo at the top there doesn't really come across very well. It's called Sepsis Research Feet, the Fiona Elizabeth Agnew Trust. And this is a group of relatives. So these are not primarily patients or survivors. These are relatives of people who've lost their lives to sepsis in critical illness, in critical care. And I think it's quite relevant to your field that we have been studying a problem that has many similarities for 4 decades now in critical care medicine. Sepsis is a very complicated, poorly understood disease, despite lots of investment in sepsis research for at least 40 years. I think what we have learned, some of it is directly translatable to MECFS. And the single most important thing is applying the scientific method, which we have done patchily in the field of sepsis. You might expect that medical scientists would always be very scientific, but that's not always been the case. Like everybody else, we have been susceptible to things that just sounded like a great idea, and because we thought we understood the biology, they just had to work, and they didn't. So we have tested in the field of sepsis with what we call phase 2 and 3 clinical trials. These are the advanced phase trials where you've convinced an ethics committee that there's enough evidence that it might be a good idea to safely give the drug to humans, which isn't usually that high a bar, and certainly is an absolute requirement before you do anything like give a drug to lots of patients. We've not just done that, but acquired what are literally tens of millions of dollars each time to do a trial. Like pharmaceutical company-led trials, sometimes in the hundreds of millions of dollars. We have done that 121 times for different treatments for modulating the host response to sepsis. Would anyone like to guess how many of those revealed that the treatment was effective? There's a bit of a hint. It was none, literally none. 40 years, we found nothing. So I think one conclusion we can definitely draw from that is we don't understand the underlying biology. We need new ideas and new techniques. So either I've got to be cleverer than the people who came before me, and I've met quite a lot of them, I'm not, or I've got to have new techniques, and genetics is the one that we applied. And so before this photo was taken, this is at the start of the COVID outbreak outside our ICU, showing nurses getting ready to go into the danger zone. And at the time, it did seem very dangerous to care for patients. We already had a study in place recruiting patients with sepsis and other forms of critical illness in intensive care. And the very first inclusion criterion was emerging infections like SARS. And we primarily recruited in the United Kingdom because the UK is very well set up to recruit patients in hospitals. But we are active now in all of the countries shown in dark blue on the map and set up in the light countries in light blue. And it was always intended to be an international study because in order to see genetic signals robustly, as you all know, you need lots and lots of patients. And thankfully, the conditions that we're looking at in the genomic study are relatively rare. So we now have 19,584 patients recruited in the UK to genomic, another 3,500 from other countries. That has been enough to see some quite strong signals, which I'll tell you about just now. So this was the first result. This graph, I'll show you a few of these. It's called a Manhattan plot, familiar to many of you. And it shows the variants we've looked at across every chromosome. So I should use the pointer here for people online. Across every chromosome of your genome lined up from 1 to 22 and then X at the end. And what we've plotted on the graph is for every one of 5 million genetic variants, how confident we are that it's really different among people who are critically ill with acute COVID-19 in intensive care against compared to the population. So it's the p-values that are plotted there. So a p-value, I'll mention again later, is a kind of standard technique in science to quantify how certain we are about a result. It's formally the probability that you would see that result by random chance alone. And the reason, obviously you want a small chance of seeing the result by chance alone. The reason that numbers are, the graph goes up, not down, is that we've taken the negative log 10 of the p-value, which just makes it easier to plot. So that's the number of zeros before the numbers start in a very small number. So this hit up on chromosome 3 as a probability of occurring by chance alone of 0.0000, 27 zeros. So pretty unlikely, but because we have 5 million different questions, there's quite a high chance we'll see chance results. So we draw the threshold for significance at this red line, which is 5 with 8 zeros behind it, so it's already quite small. Okay, so in this first result, we got this within 5 months of the start of the outbreak, and we found several genetic associations that looked like they were significant, that is, met our red line threshold. And then we tested them in 3 other control groups to be sure that they were real. And not all of these survived, but 4 of them did. And all 4 of them pointed one way or another to a potential therapy. They're the 4 actually by chance, they're the 4 along at the right-hand side here, OAS1, DPP9, type 2, and FR2. So they all pointed to biological pathways that gave us clues about how to treat the disease. And that was the intention all along, was that we'd try and find something that pointed us towards a treatment. And our particular favourite, which I'll come back to later, was this gene type 2. We've since increased the size of the study. So that first graph was with 1,660 patients. I would not expect that we will see a signal with that many patients with long COVID. It's a much more complicated problem, I think. But this graph shows the results of the same kind of analysis with just over 7,000 patients. And now we're seeing 23 independently significant genetic associations. This study, the second time, actually, again, this is about 5 million variants plotted, but rather than just looking at those 5 million variants, we actually sequenced the entire genome of all of these patients, which might in future find new findings that we wouldn't have found by any other means, and probably in this graph there are a couple that we wouldn't find by the standard cheaper technology. One of them is this gene, interferon alpha 10. So one of those spikes on that last graph is a genetic variant right in the middle of the gene that encodes this protein. So the gene provides the DNA code that is read by your cells to make this sort of greenish blue coloured molecule in the middle. And it's a signaling molecule called interferon alpha 10. And it's shown here, the protein is shown in complex with this receptor. And the genetic variant that we found is really quite likely that we understand the mechanism, which isn't always the case for a genetic variant. It changes this single amino acid, the single chunk of the protein, and replaces it with a different one. And the prediction from the structural biologists is that that would disrupt that ring at the center of the molecule, causing it to fold differently, probably leading to a completely non-functional molecule. So it gives us, I think, quite a lot of confidence about the direction of effect as well as the molecules being affected. And we've got another example here where a single amino acid change in the middle of a protein is associated with worse disease. And again, it breaks a key function of the protein. In this case, the protein is called PLSCR1, and this amino acid prevents it from interacting with the big blue molecule, which is another protein that it's dependent on for its function. So again, a broken protein makes you more sick with COVID, and the direction of effect is fairly clear. That's not the case for all of the variants we found, and in fact, in many cases, we can't know which protein the genetic variant is affecting, because it's not in the middle of a protein structure. It doesn't change the structure of the protein. It just changes something else. We don't always know what that is. And now, just to keep you bang up to date, we have in our most recent analysis, again going back to the cheap microarray technology, where we only read the differences rather than sequencing the whole genome, and we have 41 genetic associations. And we've reported this. We put it online immediately when we got the result, and we're sure that the analysis has been done properly. We're really just at the beginning of working out what the therapeutic implications might be for all of these in acute COVID. So what can you learn about ME/CFS genetics from that? I think the key thing is that the design of the study that we've done here, the results of which I'm telling you, really had an enormous impact, I think, on what we found. This shows our study compared with all of the other genetic studies that were done in the world for COVID. A lot of them are population genetic studies. They already had hundreds of thousands of people's DNA, and so the number of cases in the other studies for many of them is actually greater than the number of cases in our study. But we only study people who are critically ill with COVID. So they're extremely sick. So if you're going to look for people who are different from the rest of the population, that's where you're going to see the biggest signals for differences. But there's an extra component to it. The people who develop critical illness with COVID do so in a second phase of the illness, which is really clinically quite apparent, and there's population evidence to support this. They have been through a phase where you have the virus, you have lots of virus circulating, you have a fever, you feel generally unwell. It's an experience almost everyone in the room, I'm sure, has had. And then after that, they develop breathlessness because something else happens. And we know that it is a steroid responsive inflammatory process affecting the lungs. We know it's steroid responsive because in the recovery trial, we found that steroids were an effective treatment. And in fact, it's responsive to other immunosuppressive therapies. We don't really know the mechanisms, but we know that that's a second phase of the illness. And in fact, there are signals that suggest that the same treatments that are effective late in disease are potentially harmful early in disease. So the fact that we only study the patients who have lung injury with COVID means that we studied a group of people who all had, or not all, but the vast majority of them had the same biological process driving the illness. And that I think is why the genomic study, which is the green line in the middle, is the big one, with many fewer patients, contributes much more to our understanding of every significant association we have. So the bars are, the height of the bar indicates the weight given to our study when you combine all the studies together, so that how much of the signal comes from our one. And you can see that for every single significant finding, the vast majority of information came from our study. And that's because of study design, I think, rather than size. So I think that's probably quite instructive for the design of long COVID studies. Here is one of our findings from the first paper. I mentioned the type 2 gene that was our favorite. This is the reason why we can do a kind of analysis which Chris is expert in. So with his guidance and the help of one of his recent students, we performed this Mendelian randomization analysis. We looked at from other studies, not from our genetic study, but from other studies of cells in dishes. We know that there are a bunch of genetic variants, changes in the DNA sequence that change the amount of the type 2 protein your cells tend to make. So we can put that on this axis. So line up every one of those 20 odd variants and say, what effect does it have on how much type 2 you produce? And then ask, what effect do the same variants have on your chance of becoming critically ill with COVID-19 on this axis? And if the amount of type 2 that you produce affects your amount of your risk of severe illness with COVID-19, you'd expect to see a straight line going up there. And there are formal statistical methods to test it, and the conclusion of our most recent analysis for this suggests that it's very strongly significant. We got this signal straight away in the first analysis. So that's evidence that supports a causal role for type 2 in COVID-19, in severe acute COVID-19. It doesn't prove it. There are lots of other potential explanations. And in fact, this particular story is a little bit more complicated because there's another variant highlighted by the red cross at the bottom that actually changes the sequence of the protein of type 2 and probably has its effect in the opposite direction. So we don't really know that this is true, or rather, we don't really know that more type 2 causes severe COVID-19, at least not on this evidence alone. But it does suggest a direction of effect. It suggests that more type 2 signaling is harmful. And on that basis, we predicted that a treatment that inhibits signaling of type 2 should be beneficial in this later phase of severe COVID-19. And so we made that prediction in the first paper that when we reported it in 2010. I had the privilege of being on the steering committee for the recovery trial. And we'd already discussed including this drug that inhibits type 2 called baricitinib in the recovery trial. There was other evidence in support of it from some small inconclusive trials. And on the basis of this genetic hint that it might be an effective treatment, we added it into the recovery trial. And I think it's important to remember how lost we were trying to find treatments for COVID in 2020. I think it felt like we had no evidence to base any of our decisions on because it was a completely new problem we hadn't seen before. I think actually that's how it felt because it all happened very quickly. If I look at the field of sepsis research, we are equally bewildered. It's just that we've been reading about it for so long, we don't recognize that we're equally bewildered. Anyway, we put baricitinib into the trial, and this is what happens if you randomly assign people to receive baricitinib or usual care. If they're already sick with COVID and already in the other treatments, dexamethasone and another immunosuppressive therapy. So this is how you know if a treatment works. I'm sure you're all familiar with this. You can't know if treatment works unless you try it. This is how you try it. You have to randomly assign people to get the treatment, randomly assign some other people to not get the treatment, see who does better. That is the only way that I know of to know that a treatment works. I'd love it if someone else had another way, but it's the only way I know any of the treatments I use anywhere in clinical practice are effective. And for baricitinib, if you're randomized to receive it on day 0, you're on the red line, and then your chance of dying, or being dead 28 days later, is lower than if you were in usual care. And we can test that statistically as well, and it meets our threshold for significance. So baricitinib is a third effective treatment for COVID and was in part inspired by genetic evidence. I think that's the first time that this has happened, and that we've gone from a genetic signal to a treatment, certainly in infectious diseases and intensive care medicine. The judgment of whether to put the drug into the trial in addition to the other treatments was exactly the problem, I guess, that the ethics committee are having with Doug's trial here. We have to make a judgment about how good is the evidence, and is it really safe to, is it really a good idea, a good enough idea to take the risk of randomizing people to receive that treatment. We had a discussion about that, and there was a rheumatologist who joined the discussion, who is from a group of doctors who use these drugs all the time. These are drugs that are used to treat rheumatoid arthritis. And they said that adding baricitinib on top of the other effective drug, tocilizumab, which is another immunosuppressive drug, is something that they would literally never do in rheumatology, because it would be considered to be very high risk. In part because we had the support of the genetic evidence, in part because the risk of death was so high from COVID, and we were literally seeing people die every week. We decided to include baricitinib on top of tocilizumab, which means we'd be able to find out if it was an additional new therapy rather than just an alternative, which obviously wouldn't be quite as useful. So I argued quite strongly for that, based on what I thought was quite a dispassionate reading of the evidence. And I felt confident that I'd done my due diligence and really thought about it. And then 1 week later, I was treating a patient in the intensive care unit who was admitted, who was already on dexamethasone and tocilizumab, was in the recovery trial, and was randomized to receive baricitinib. And I have to confess that when it came to actually writing it on the patient's prescription chart myself and looking her in the eye, I did second guess the decision making. It felt quite different from trying to make a dispassionate scientific assessment. I probably wouldn't have mentioned that story if we hadn't found out that baricitinib was effective. Okay, so I'll finish by trying to synthesize this. So I think we've applied the scientific methods to try and find solid rock evidence to support our perception of COVID. I think we have to then build some sort of synthesis of what we think that's telling us. But here I'm moving from hard evidence, which is what you should all base all of your thinking on, into a bit of conjecture, because I don't have hard evidence for everything here. But this is our sort of broad model of what's happening in patients with COVID. I mentioned that initially there's a phase of illness where you have lots of virus around. In red here, I have plotted an imagined series of biological processes, molecules interacting with molecules in your body, that are initiated by the virus. If it's in red, it's initiated by the virus. And I've deliberately chosen the vague term bad for anything that's above the line here. You might, if you wanted to put a really precise definition of that, say something that decreases your chance of being alive 6 months from now would be bad. And broadly speaking, the responses in blue, the processes in blue rather, initiated by your own immune system and your body, are probably good. But you have to keep an open mind. Some of those things are probably not doing any good. Some of the things that the virus starts off in the process might not do you any harm, or might even increase your chance of survival. So that's the initial phase of illness. That's the illness that I've been through with COVID, where a bit of a fever, felt quite unwell, got better. If people don't get better, and again, I'm talking about acute COVID here, not long COVID, maybe they have more virus. So maybe if their host immune response isn't as strong, more virus at higher peak, or maybe they have the virus around for a longer time, or maybe both. And we know some of the mechanisms that underlie that. So 3 of our genetic associations, the 2 that I showed you with molecules, where I said the molecules are broken, I think we can be confident that the function of those molecules being broken increases your chance of becoming critically ill. And we kind of know enough about them to think that probably have an effect in controlling the amount of the virus that you have circulating. These are elements of the immune system that have been known since 1957 to be critical for controlling viruses, and if they're broken, and I think it's likely you'll get more virus. So that's probably where they have their effect. But then in some people, there's a second phase of the illness, which we, I think, have good evidence to think is driven, at least in part, by the host immune system. And the best evidence, of course, comes from the fact that if you give people steroids, it gets better, or rather, they don't die. Same is true for tocilizumab, the other drug that I mentioned, and baricitinib, the one that I've been telling you about. And actually, there's probably a signal in the opposite direction for treating people with interferon. Interferon is this signaling molecule in the immune system that controls viruses. It's the canonical antiviral signaling molecule in the immune system. But if you give people interferon in a randomized controlled trial, when they're in hospital with COVID, so probably in this later phase, they don't do better and probably do worse. There's a big trial, the WHO Solidarity trial, that found that. So again, probably more interferon signaling here might be bad. And then we have a range of genetic variants, and just for space, I've only put 5 of them up here, that probably, and again, this is a bit of conjecture, but because of their roles in biology, we think they probably have their impact in this phase of the illness. And it includes type 2 and JAK1, both of which are inhibited by baricitinib, and a few others, including a couple, factor 8 and this platelet-derived growth factor ligand, that are implicated in blood clotting, which suggests that abnormalities in the clotting cascade might have a role in acute COVID. And that fits with autopsy evidence and some of the physiological evidence from acute COVID. And finally, the second phase of the illness gives way clinically to an even later phase in the really sick patients who are in the intensive care unit, where they essentially converge with patients who've been critically ill for any other reason, because by the time you've been in an intensive care unit on organ support for several weeks, your problems are mainly not related to whatever got you there, but to the complications of organ support and to how sick you've been. Okay. So I'll finish there by telling you what we intend to do in acute COVID and sepsis and critical illness to translate these findings from genetics to clinical practice. So in some cases, and I think we probably have to accept there was quite a lot of luck involved in that we went straight with type 2 and baricitinib into large-scale trials. In some cases, we get a clue that's so good that we can do that, but mostly we have a lot more work to do. We can stratify patients, and I think this will be important in sepsis, and I think it'll be important in long COVID and NECFS. And genetics offers us a bit of a tool to do that. We can separate patients into subgroups based on our ideas about who's got different symptoms or different mechanisms of disease revealed by clinical data. And then we can test whether those subgroups are really biologically coherent. So we had the good fortune in acute COVID to have a group of people who were very similar to each other, the same mechanisms of disease. They actually looked clinically very similar to each other. And we can probably test whether our ideas about how to substratify a population are correct using genetics, using sort of feature genetics that it sometimes acts a bit like the drug therapy. So for example, we can either use the drug effect to do that or genetics. So we can either say this group of people the drug worked in and this group of people in the same population didn't. Or we can say this group of people have this genetic signal and this group doesn't. They're the two ways I can think of that you can really test whether two subgroups within a disease are different. But of course, your ability to tell whether the groups are different depends on how strong your signal is. And the p-value, which is the concept I explained earlier, the probability you'd see a given result by chance, for a result from recovery for baricitinib is 0.023. So for context, 0.05 is the standard threshold for whether you accept it or not. And for our strongest hit in the genomic study just now, in the unpublished data I've just shown you, the p-value is shown, there are 249 zeros there. And that's, of course, because it would be profoundly irresponsible to keep doing a trial after you knew the answer. It would be very unfair to randomize people to not get a drug you knew works. But we can't stop remaking this discovery. Every time we run the analysis, it gets stronger. We would like to, because we want to find more, but we can't. And that means we've got this untapped resource of what we call statistical power that will enable us, I think, to design clinical trials in silico. And then for the lung, we're developing technologies for testing drugs by administering tiny doses of drug to little bits of lung in critically ill people with the actual disease that we're treating. So I mentioned the recovery trial, that was led by Peter Horvey and Martin Landry, I think are famous enough not to need to be acknowledged. My collaborators and team that ran the genomic study really delivered the results that I've been telling you about, so particularly Arola Padacastanera, Sericlohisi, Lucia Claridge, Andy Bretherick, and Conrad Rolick, who all sacrificed their sleep and sanity to get the work done as fast as possible. And of course, there are two other groups of people who really made the study possible. People I showed you in the first slide, donning PPE to go into what was thought at the time to be a very dangerous place, the intensive care unit for COVID people. Their names are shown here. 1,440 research nurses and clinicians who approached patients and recruited them into the study during the unimaginable circumstances of the pandemic. And of course, the patients and their relatives who, at a time when it was probably the most frightening time in their lives, thought to contribute to a research study to help other people at no possible benefit to themselves. I'll stop there. I think I've left time for questions.
Oh, fantastic. So there will be lots of jazz hands here and online. And I just want to thank you for giving such a clear presentation on what has been of profound importance to so many people. So if we can have questions online and in the room, please. It's Steve first.
Hi, Steve Gardner from Precision Life. I was wondering, firstly, what your control strategy was for the trial.
Yeah, so I skipped over that. So it's messy to start with. So we recruited cases and then found controls from existing studies. And UK Biobank was the one that we used for the first.
With a COVID diagnosis?
No. So at that point, I mean, 5 months in, very few people in UK Biobank, thankfully, I guess at that time, they've all had it now, but very few people had COVID. And also the information, whether or not they've been exposed to the virus, is unknowable and when it was knowable, hadn't percolated through the system. So broadly, they were population controls. And although we removed anyone who was known to have had COVID, there were a whole bunch of people in there who probably did have, not just who'd had COVID, but also who would have had the chance of becoming critical, who would have been expected to get critically ill if they got it. So that decreases our signal. It makes our control population more similar to the cases, but it doesn't throw out on any signals that we find. We also then, in subsequent analyses, confirmed using the Generation Scotland database and 100,000 genomes population studies. And we recognised, so my colleagues, a particular statistical geneticist in this university who constantly slags me off for designing a study that recruited cases and not controls. We did recruit controls, and there are 15,000 of them, very generously contributed members of the population. And we're analysing those data now, but it took a while, so it's not finished.
I think Joshua had I'm sorry.
Thank you, excellent talk. Can I ask about so you had very, very strong genetic signals from your on your Manhattan there. What were the allele frequencies like for the lead SNPs? Are these rare variants?
Yeah, some of them are. So interferon alpha 10 is the one that's below 1%, which I think is the one that we can realistically claim whole genome sequencing got for us, that microarray genotype we wouldn't have, because we would just have filtered it out. And they range, I mean, I can't remember the numbers on the table. It's on medRxiv right now, that unpublished data that I showed you. But they range from there to well over 50% for the risk category.
Miriam, I'm going to take two after Miriam online, and then Steve.
I'm trying to get your most recent data. What is the proportion of the patients who have been vaccinated that's still including not any evidence and that changes?
Yes, so it's getting interesting. Yes, so we thought carefully about what are the routes to the intensive care unit with severe COVID. And I think there are, my model, I've got two. One is you don't fight the virus well enough. The first phase. The second is you're somehow predisposed to having this inflammatory response in your lungs. The third, that you have been given the vaccine and have failed to mount a response to it, I think is now going to be a feature. Now, I don't think you need to persuade this audience vaccines are really terribly effective. They're a great idea. And as a consequence, I have not seen a patient in the intensive care unit this year who had COVID as their primary reason for being admitted. Now, the other thing is the virus has changed, but we've got now a very well vaccinated population. I don't think we're going to be able to tease apart that signal, but we are comparing the waves of the virus And we have, so in the first plot, I showed you 4 genes that I said, predicted therapeutics targets. One was OAS1, and that sparked for about a year and a half tremendous excitement in the field of virology and lots of in vitro experiments, because it's a gene made by your cells that controls viral replication in cells, but not all viruses, and not the other coronaviruses that infect humans, just SARS-CoV-2. And then in the second analysis, I didn't point this out, but that signal had gone away. So wave 1, strong signal, wave 1 together with wave 2 and a bit of wave 3, really no signal at all. And it's the only gene that did that. Now I've been cautious that we summarize that, because we've tested it whether it was significantly different, wave 1 to wave 2, and it wasn't quite significant. So we're not really sure that it really changed. But we have asked a colleague, Sam Wilson of Glasgow, looked at the effect of that gene on restricting the virus from wave 1, wave 2, and wave 3. And by wave 3, the virus changed. The Delta variant was not restricted as much by that feature. So we do see some of those signals, but I don't think we'll have enough data to see on antibiotics.
So these are legacy Q&As. So
Okay.
Steve.
That's an answer. Thank you.
Okay.
Okay. Any questions in the room or online? Yes, one.
Just wondering whether you'd be able, so the ones that you mentioned were more involved in fighting the virus or the interaction with the virus mostly. Do you find variants that were more related to the, like, similar to the sepsis response, similar to the host response?
Yeah.
Yes.
Yeah, so those that blue search, that's really where I get interested, and it's not because I'm just focused on the questions I like. That's because I think I can't realistically expect to find or to try and deploy a therapy that has to be deployed before I meet the patient. And most people with critical illness and COVID present, the first presentation to the healthcare is when they get sick enough to go to hospital. So that's where I'm interested, and we are finding genes that all we know about the genes is whatever has been written about them before. But for the ones that are in the immune, relevant to inflammatory actions of the immune system and clotting, I think that's where they're active. We have found now, quite late actually, in quite sort of buried deep in the sort of less strong signals, we found a significant association for the 2 proteins that are known to be directly affect the virus getting into the cells, ACE2 and CMPRSS2. They're there, and that was kind of proof of concept, but didn't really excite us.
Okay, so if there are any further questions online, then please put them in the Q&A, and I'll ask Kenny if he can respond to them in time. But fabulous, and thank you very much for everything that you and your team have done over the last 2.5 years. So it is yet again an immense privilege to have with us today Steve Gardner from Precision Life, who is going to tell us about their incredibly interesting method to identify DNA variants that together in combination predict long COVID and ME and anything else you wish to talk about today. So good, Steve, and Steve's team is with him today. So if anyone wishes to talk to his team, then please talk. Oh. So I'm not sure that is showing online.
Okay.
Is it? No. Here.
Right. Oh, good online. Yeah, excellent. Thank you.
Well, the pleasure is all ours.